Hi, everyone. My name is Ethan Lee, and this summer I joined Dr. Jane's lab at Washington University in St. Louis, and my project was contributing to the generation of a multimodal pediatric kidney atlas. All right, so the kidneys are vital organs that filter out blood for waste products, regulate fluid and electrolyte balance, and also control blood pressure through hormone production. And recently, Dr. Jane was involved in the HuBMAP collaborative publication, an atlas of healthy and injury cell states and niches in the human kidney, which mapped the human adult kidney in single cell resolution. So this atlas has been super important as it identified rare and previously undescribed cell states, provided insights into disease mechanisms, specifically altered cell states, and also, I think most importantly, provides a strong foundation for future researchers to build off of. So this summer, I contributed to the building of a pediatric kidney atlas, which is a similar project, but designed to map the pediatric kidney and track its development at single cell resolution. So the nephron is the kidney's functional filtering unit, and it forms during a process called nephrogenesis. Although there have been efforts to map nephrogenesis, no one has produced a pediatric kidney atlas focused on postnatal development. So at the top left of this slide, you'll see the general timeline of nephrogenesis, and below it, the diagram of nephron development during nephrogenesis, and you'll notice that it concludes before birth. So our project's primary aim is to map pediatric kidneys from neonates through teenagers at single cell resolution. And this is important because it'll provide insights comparable to the adult kidney atlas, with the added benefit of us gaining a better understanding of post-nephrogenesis development. So in addition, this is especially critical given the serious consequences of pediatric kidney disease, with it being a large contributor to organ failure in kids. And we currently lack the comprehensive developmental framework needed to better treat these conditions, and this atlas will help address these gaps to better inform future diagnoses and treatments. So the foundation of this atlas is single cell analysis. Single cell analysis is critical for any sort of atlas, as it gives you insights into cell heterogeneity in tissue. So this diagram shows it pretty well. You can see that with bulk RNAs input from a tissue sample, the RNA expression profile you get is an average of the heterogeneous types, and you lose the information that is critical for an atlas, specifically differentiating between the different cell types in the tissue. So this is a diagram of the overall pipeline of developing this pediatric atlas, which I'll go over briefly. So tissue is harvested either through nephrectomies, biopsies, or from deceased donors. The tissue is then processed at the single cell level for both nucleic RNA and for ATAC. So nucleic RNA is telling you what genes are being transcribed into RNA, while ATAC is telling you what chromatin areas are accessible for each cell. So when repeated for thousands of cells, you end up with massive amounts of data, which you process computationally before then visualizing with a graph called a HuBMAP. So samples are collected either through kidney biopsies or full nephrectomies. These samples are first embedded into OCT, which supports freezing, before then being sectioned off by a cryostat machine. These samples then undergo nuclear isolation before then being sent off to 10x genomics for multiomic RNA and ATAC. So 10x's processing generates a massive amount of data. For the pediatric atlas, we have around 150,000 cells and 30,000 genes. So two popular single cell analysis tools that we used are Seurat, which has tools for RNA analysis, and Signac, which focuses on ATAC analysis. So to the left, you'll see a draft of the overall pipeline for this analysis. And I won't go super into detail, but the general gist is that you're given your data in a massive matrix where genes are rows and columns are cells. You filter for quality cells before then plotting in a high dimensional space. And you then reduce the dimensionality while keeping important variability. And then in this reduced dimensional space, you can cluster cells based on their transcriptomic similarity. And this is represented as the different colors in this graph. So this graph is finally generated by, again, compressing the dimensionality to 2D for visualization purposes. So in this pipeline that I described, it will cluster based off their transcriptomic similarity, but it won't tell you the identities of these clusters. So I worked on labeling each cluster of a subset of this atlas called the distal epithelium, which includes the thick ascending limb, the connecting tubules, and finally the collecting duct cells. So this process involves identifying the uniquely strongly expressed genes in each cluster, and then cross-referencing with previous data to determine each cluster's identity. So something that I wanted to point out that's interesting is that you can see in the UMAP kind of reflects the spatial organization of the nephron itself. So for instance, the thick ascending limb is directly next to the connecting tubule spatially, and that's also the case in the UMAP. And the connecting tubule is also next to the collecting duct cells, which is also the case in the UMAP. So following the labeling of this object, I began attempting to analyze some of the developmental pathways that could be present in our data set due to the variety of age ranges that we captured. So the first tool that I applied is called Slingshot. And basically, given a group of cells that have some developmental relationship, that is like progenitor cells or end state cells, you can plot a potential pathway for development. It is important to note, though, that this is dependent on user inputs, and it's only meaningful when you have a group of cells along a trajectory pathway. So I focus specifically on the development of the macula densa, and the macula densa is a group of specialized epithelial cells located in the thick ascending limb of the loop of Henle. It senses sodium chloride in the tubular fluid and is positioned appropriately, as you can see in the middle image, to signal to arterioles to either constrict or dilate to maintain stable filtration, while also being involved in the renin pathway. So while it is a control center for the nephron, not much is really known about its development. So the figure on the right is from a paper focused on nephrogenesis, and you can see that while it has the general splits down, it doesn't determine how the macula densa forms, such as like whether it arises from the thick ascending limb or from some shared progenitor between the macula densa and the thick ascending. So in the pediatric atlas, Slingshot was an applicable tool. We had identified a cluster of interest, which is the gray cluster in the middle on both plots. We also had enough macula densa cells having more than 1,300 and from all age ranges. You can see that the middle cluster is labeled MD/cortical tau, as it's expressing markers of both macula densa and the cortical tau. So this could either be a transitionary cluster between the two groups or a potential progenitor cluster. So I ran Slingshot on it, first resulting in the plot on the left, and then I subsetted and reattempting, giving the plot on the right. So that was important because subsetting in single cell analysis is important because analyses can be thrown off by clusters such as damaged cells that weren't identified, or just made more complicated by irrelevant clusters, which in this case is cortical tau B. So with the Slingshot analysis and other single cell tools, I began searching for relevant genes to the macula densa development. This is an example of like an ideal gene for development with one caveat. So you can see that it's primarily expressed in the tau with this graph on the left, which is called a dot plot. So the larger the circle, the higher the expression, basically. And in the middle, which is specific to the thick ascending limb region, you'll see that there's high expression in the potential progenitor cluster, high expression in the cortical tau A, and basically no expression in the macula densa. And then further to the right is the trajectory plot. So I had set the start point of this trajectory as the cluster in the middle, and one trajectory is to the right, basically going towards the macula densa, and the other is to the left towards the cortical tau. So this plot up top is basically showing the gene expression as you progress along these two trajectories, with the yellow one being the macula densa trajectory. And you can really see how this gene expression drops off the macula densa. And the caveat that I mentioned earlier is that this is a long non-coding RNA, so it'll be like very difficult to test in one lab. So the next tool that I applied to this object is called RNA velocity. So this is a really interesting tool to me, at least, and it's a lot more unguided than Slingshot. So based on the ratio of spliced to unspliced messenger RNA in itself, you're able to estimate whether a gene is being upregulated or downregulated. And across thousands of genes, you can create a vector in a high-dimensional space for that cell, with arrows pointing towards that cell's next transcription space. So what that looks like, at least for the macula densa / cortical tau region, is this, and you can see that there's arrows going from the middle potential progenitor cluster or transitionary cluster towards the cortical tau A, and a few arrows going towards the macula densa. So while I was limited by time, I do want to continue doing downstream analysis, specifically on this same area. So one tool that I've started applying already is called CellRank, and that basically further builds on RNA velocity data to predict initial cell states and terminal cell states, rather than just like the immediate next transcriptional state of the cell. So another tool that I want to apply is Cell Oracle, which allows you to validate in silico the impacts of a gene knockout, which would be super helpful for all of the genes that I came up with this summer. And more long-term, I want to assist in the validation of like these findings in wet labs, specifically with organoid model knockdowns. So in conclusion, this summer I contributed to the trajectory modeling in the pediatric kidney atlas in the gym lab. So I wanted to give a special thank you to Kaka, my bench mentor, and Dr. Jane, who helped me come up with like basically all of my presentation and gave me a lot of guidance this summer. And thank you to the Jane Lab as a whole, HuBMAP, for giving me this opportunity, and to the Pediatric Center of Excellence in Nephrology for teaching me all about the kidney and giving me opportunities to shout it this summer.
All right, good job. That was well done. All right. Does anyone have any comments or questions for Ethan? Go ahead.
Yeah, I just wanted to say good job. I loved your presentation. I was wondering how much you knew about nephrology or the kidney before going into the Jane Lab.
So I had taken a physiology course before, and we had a unit on the kidney, but definitely nothing as like in depth as was like required for this research, which was really good because I got like to learn about the kidney in like a hands-on way. And also I learned about like the clinical aspects of nephrology.
Totally. It goes much more in depth. I was an intern in the Jane Lab a couple years ago and I
Oh,
worked with
okay.
Becca. Yeah,
Oh, wow. Okay.
I
Wow.
did. Tell her I said hi.
Yeah, I will.
Thank you.
Good question. Anyone else have any questions or comments?
I did have a little question. With your diseased samples, was it just CKD you were looking at, or were there any other pediatric kidney disease you were looking at?
So I think there were CKD samples. I wasn't too certain about like what disease samples they are going to be included in the final atlas, because I think that's a topic that's up for discussion right now. But yeah, I'm not too certain about like which disease samples would be included, to be honest. Other than CKD.
I only asked because I used to work with like ARPKD, which is a recessive polycystic kidney disease in usually young people. So,
Yeah.
but thank you.
No problem.